In this video, I'm going to discuss selective and differential media. So typically when we get a culture that's sent down to the hospital from a patient, we need to be able to differentiate or distinguish between the microorganism that's actually causing the infection versus the normal microorganisms that just live in the body, and even compared to normal contaminants that you might find in those types of samples. So there is selective and differential media that allows us to get a diagnosis a little bit faster. So I'm going to go through just 4 types of media that are pretty commonly used in a clinical setting, starting with this one. So this one is blood agar. Blood agar is, it contains sheep blood, and that's the most common type of blood agar you have. Sometimes different substances can be added to the blood agar, but this is just simple sheep blood agar. Blood agar is useful because it allows us to distinguish microorganisms based on their ability to eat the blood cells, and that's known as hemolysis. Hemolysis. Lysis means to break apart. Hemo is talking about the blood, so the ability to break apart the blood. When bacteria is put on this medium, it's going to either not break down the blood at all, and that's known as gamma hemolysis. If you see gamma hemolysis, you won't see any breakdown of blood around it. You're just going to see pure colonies on the surface with no disruption of the color of the actual agar. There can be partial hemolysis, known as alpha hemolysis. In alpha hemolysis, you're going to see a greenish color around. If you recall the breakdown of hemoglobin from possibly previous classes, it's broken down into biliverdin and then eventually bilirubin, which has a yellow color. So biliverdin has a green color, that kind of moderate breakdown or digestion, that medium hemolysis that you would see from some microorganisms, that's alpha hemolysis. The other type of hemolysis would be beta hemolysis, and beta hemolysis is complete hemolysis. You would see a yellowish hue to the microorganism in the agar surrounding the microorganism, or you might be able to pick it up and hold it up to the light and be able to see all the way through. So because it can differentiate bacteria based on the hemolysis, this is known as differential media. It is not selective media. Pretty much everything that we would like to grow will grow on here, with of course some exceptions, but most microorganisms are going to grow just fine on this. The next type of medium is just a nutrient medium. Nutrient medium is just agarose with added nutrients to it, and this grows a wide variety of different microorganisms. Nutrient medium, TSA is another type of nutrient medium. This is just your general go-to medium to ensure that you have some type of growth. It is not selective, nor it is differential. This next type of medium is Mcconkey agar. Mcconkey is used to grow gram-negative microorganisms. It is both selective and differential. It's selective because it selects against gram-positives. And this is used for enteric pathogens. So the enterics, the ones that live in your gut and in your digestive tract, those are, there's gram-positives and gram-negatives in there. Typically in your digestive tract, the gram-positives are going to be the good guys. Those are the probiotics and the ones that are going to protect you from these types of infections. The gram-negatives are usually the bad guys. So when we grow digestive sample, fecal samples, that type of microorganisms on this plate, it's going to kill off the good guys because we don't care about the good guys, and it only grows the bad guys. So that kind of reduces the number of microorganisms that we're looking at, so we can identify what's causing the diarrhea or what's causing the abdominal upset and that type of stuff a lot faster. So it's selective because it contains crystal violet and bile salts that are going to select against those gram-positives, and then it's differential based on lactose fermentation. Some enteric pathogens have the ability to eat lactose. It's known as lactose fermentation, and some don't. So if they do have the ability to eat lactose, they're going to turn pink in color. If they don't, they are going to just grow the natural color that they normally grow. So selective and differential, and this one kills off the gram-positives, so it selects against gram-positives. It only allows gram-negatives to grow, so that it selects for gram-negatives. And then it's differential based on lactose fermentation. It'll grow a different color based on its ability to eat lactose. This medium is collagen nalidixic acid, Cnaagar, and this one selects for gram-positives and selects against gram-negatives. This one is commonly used for skin infections and wound infections. The reason why we use this is because on the surface of the skin, there's some gram-negatives that we don't care about. We want to grow the gram-positives because typically skin infections are going to be caused by those gram-positive microorganisms. So it's selective because it selects against gram-negatives, so it kills off the gram-negatives, and it selects for gram-positives, so it only allows those gram-positives to grow. And of course, there's exceptions to all of these. Some gram-negatives will grow on here, some gram-positives will also grow on Mcconkey, but for the most part, it does a pretty good job at reducing the number of microorganisms that we're looking at so that we can get a faster diagnosis. This one usually is accompanied with blood. So usually you have CNA with blood agar. This one is just CNA on the surface. It is not differential unless you were to add blood to it. The blood would make it differential. So these are the 4 types of media, and I'm going to show you some results of this media and how microorganisms grow in the media in just a little bit.